Hello, everybody, and welcome to today's talk in our Evolution and Ecology seminar series. My co-organisers and I are very excited to have with us today's speaker, Professor Anne Charmantier. Anne is a professor in genetics and evolutionary ecology at the CNRS, the French National Centre for Scientific Research in Montpellier. She obtained her PhD from Montpellier, after which she held 2 postdoctoral fellowships in Oxford, before finally returning to Montpellier to the Centre for Ecology and Evolutionary Ecology. Anne's research focuses on the effects of behavioural and life history variation between individuals in natural populations. With her group, Anne runs very exciting long-term projects that aim to understand evolution in response to changing environments, such as global warming and urbanisation. Anne's research is very multidisciplinary. She integrates ecology, evolution, behaviour, quantitative genetics, and genomics. And I'm very much looking forward to her talk entitled Great Tits in the City, from Phenotypes to Epigenomes. As usual, there'll be a Q&A session with Anne directly after her talk. Please post your questions in the designated Slack channel. You can find the link to that one in the description of this stream. And please upvote questions that you would like to hear answered at the end of the talk. So Anne, thanks again for accepting our invitation, and over to you, the floor is all yours.
Thank you, Andres. Hello, everyone. This is really a first for me. So I first wanted to warmly thank the organizers of this seminar series I've been following. I think it's really helping us to connect and to communicate in this complex situation that we're facing. This week, I was actually supposed to be in Brisbane for the ICQG, the International Conference of Quantitative Genetics, and I would have been halfway through a sabbatical in Australia. And in fact, this morning, I was chasing great tits here in my hometown in Montpellier, and now I'm talking to you all who I'm not seeing. So I'm going to have to try and imagine you nodding or frowning and give my talk in a very different perspective. But so I'm going to start with, my talk is going to be really in 2 parts. First, it's going to have quite a lengthy introduction on evolutionary biology conducted in urban environments, trying to get you as excited as I am for this emerging field. And then I will give some results about our own project in the city studying great tits. So as an evolutionary biologist, I have a major interest in studying evolution in action. That is what we call microevolutionary responses of traits across different generations. And this microevolutionary response is fueled by selection, so selective forces that are acting on heritable traits. During several decades, I'd say, it is in the plant and the animal domestication and breeding science that the best examples of microevolution were found, and also that the best progress in understanding and predicting rapid evolution was made. And in fact, it's in this field of animal and plant breeding science that quantitative genetics was developed, and especially what we call the breeder's equation here, which predicts the evolutionary response in a character based on the heritability of this trait and the selection acting on this trait. So in this framework, microevolution is really predictable. And that means that, for example, for a farmer with a cow herd, the farmer can predict the increase in the production of milk over the years based on the heritability of milk production, which is known, and based on the selection that this farmer decides to incur on the cow herd. Now, building on these plant and animal breeding programs that aimed really at mainly increasing productivity, over the last 2 or 3 decades, we've been using long-term individual monitoring programs in the wild to try and apply quantitative genetics with the aim of understanding and maybe one day predicting evolution in action in these wild populations. So if I were to ask you, what is in your mind one of the best examples of evolution in action in the wild? I think a lot of you would talk about Darwin finches. And indeed, I think it's one of the best examples we have of evolution in action outside domestication and breeding programs. So these are studies especially on the beak depth by Peter and Rosemary Grant on medium ground finches. And these studies showed that in these Darwin finches, the beak depth evolves at quite a fast rate especially following extreme climatic events such as El Nino or La Nina events. And recent genomic investigations have revealed that there is this one major locus involved, HMGA2, which determines beak size. And this gene facilitates the rapid evolutionary response in this character. Apart from beak size in Darwin finches, rapid evolution hasn't been demonstrated in so many populations, but we do have a few good examples. And I'm going to give 3 additional examples here, which are one of my favorites. So the first one is all the studies that have looked at pesticide resistance in, especially in Anopheles mosquitoes. The second example, which I often use, especially in courses, is this study by Pierre-Olivier Cheptou and his group. So these are people working in my lab that have been studying the weed Crepis sancta in urban environments. And they have shown that when this weed arrives in urban environments, it will rapidly evolve towards producing seeds that are less dispersing seeds rather than dispersing seeds that will end up in unfavorable environments. And this happens in around 15 generations, this evolution. And then the final example I wanted to illustrate here is the soapberry bug, which has evolved a morphological and life history adaptation to an introduced host plant. So one idea that is emerging from the literature is that most examples that we have of rapid adaptive evolution are in fact in response to anthropogenic change, and many of them are from urban habitats. This is a figure that was that I copied from a paper published by Mark Johnson and Jason Munchi South in 2017 in Science. And for me, 2017 is really the year when urban evolutionary biology was born. And I say this not only because of this review paper that was brilliant, but also because it was the first year where we organized special sessions in our international conferences of evolution, both in North America and in Europe, on urban evolutionary biology. So in this paper, what this figure illustrates is, so in the dots here are cities that are at least 300,000 inhabitants, and all the black silhouettes are case studies of urban evolution. Now, it doesn't mean that all these studies have demonstrated urban evolution, but they have investigated it. And the blue silhouettes here are areas where human commensals have evolved. I actually watched a webinar an hour ago by Mark Rabinet on the evolution of human commensalism in house sparrows. That was really exciting. So there's a lot of work being done on this as well that I'm not going to talk about today. So this slide is about convincing you why we are excited about urban evolutionary biology, why we think that it's really a golden opportunity to study evolution in action. And I've listed a few reasons here. The first one is that because urban environments represent environments that are so drastically different from natural habitats, we expect that they will incur very intense novel selective forces on organisms that stay around and live in the city. Now, I insist here on the expect because this is actually quite a very wide assumption that has in fact rarely been tested. And I recommend Sally Otto's review that was published in Proceedings in 2018, where she discussed how humans are shaping selection pressures. The second reason is that cities are all around the world. They are expanding at a massive rate, and they are very similar in their features in different countries. So that means that it offers multiple spatial replicates, which is ideal to test parallel evolution. And also in all these cities, the environmental axes of variation are actually quite easy to quantify in many ways. And this is because if you live in a city where you want to observe and study biodiversity, you can have access to a lot of data from your city services, which monitor parameters like air or water pollution, and that can be very helpful. Now, I also want to mention that if urban evolutionary biology is very recent, urban ecology is quite an old field. It's I would date it to about 50 years ago. So we have a lot of knowledge on biodiversity in cities, and we can build on this knowledge to establish the fields of urban evolutionary biology and target species or predictions that we want to test. The two last reasons I want to highlight here are somewhat personal reasons, and they are linked to our role as science disseminators, but also as world citizens. So you might know that presently half the human population lives in cities. And by 2050, it'll be 2/3 of the human population that will be urban. So the fact that cities are expanding is an increasing problem that is not about to become smaller, right? It's going to stay around for a long time, and it's becoming a societal environmental issue. I also find that talking about evolution to people by highlighting species that they can watch on an everyday basis is a great entry for environmental education. When I go into school, I find it much easier to talk about evolution of species that the kids can watch in their gardens. So I find this an exciting perspective as well. So all these reasons have brought us, and by us I mean a collective of evolutionary biologists working in the urban environment, to work on this edited book that came out last month. So I could not mention it today, especially because, as you may know, books are usually, the date at which a book is published is usually targeted for conferences, summer conferences. And this summer, you're not going to see our book on the shelves of your favorite conference. So I thought I would mention it. But I'm also mentioning this book because in chapter 5, I have worked with my postdoc Charmantier and PhD Odd Kizerk, who I'm going to talk more about later on, about building a figure. So this figure is a proposed framework of how we can explore evolutionary processes that may lead to adaptation and adaptive evolution in cities. But it's really applicable to other environments than cities. So the logic that I usually take in these kind of studies on rapid evolution is that we usually base our first observations on phenotypic differences. And the leitmotif graphical example I'm going to use here is the melanic and non-melanic forms of the pepper moth morphs in the UK. So Biston betularia. And this is just to illustrate our approach here. So you often start by observing two different morphs or variation in other phenotypic traits. And as an evolutionary biologist, the first question that I will ask myself is whether these two morphs are adaptive. And this is where you can start conducting selection analysis, where you confront inter-individual variation in fitness with the variation in the morphs. And then the next question we usually ask ourselves is whether this phenotypic divergence is of genetic or plastic origin. And for this, one of the approaches that has been developed most in the past, and especially before the advent of genomics, is to conduct experimental approaches, such as cross-fostering or common garden experiments. And this will tell you whether the different morphs are of plastic or genetic origin. Now, I want to say here that although this genetic arrow is linking us to the genomic area, the plastic arrow might as well, because even if the dimorphism is of plastic origin, plasticity itself can be heritable. So you might want to answer the question of genes involved, even if there is plasticity involved in the process. And to illustrate this part, which is the genomic part, I'm going to take examples from the bird literature, which is the literature I know best. So we've split this genomic part in three different types of approaches. The first thing you can do is you can target candidate genes, especially if you know candidate genes that are involved in the trait that you've been observing. So for example, in birds, there are candidate genes SERT and DRD4, who are involved in human avoidance and shyness, boldness, so personality traits. And they've shown SNP variation between urban and rural population in blackbirds, in the black swan, and in the great tits. Oops, sorry. The second way that you can approach the question of genes involved and their location is by reducing genome complexity and using methods such as rad sequencing. One of my favorite examples is, actually, this is not a bird. I don't know why I said I would take only bird studies as example. This is completely not true. So yeah, the work done in Jason Munchi South group, and especially this paper by Harris and Munchi South, where they analyzed more than 150,000 SNPs from transcriptome data in white-footed mice from New York City and from rural areas around the city. And they found multiple SNPs associated with urbanization in these white-footed mice. So they concluded that the mice in New York City are adapting at the biomolecular level to these local selective pressures that are represented in the urban habitats. And finally, if you have a big budget, you can also conduct whole genome sequencing projects. And here I'm closing the loop with my example in the peppered moth, because in this case study, a transposable element was identified as responsible for the industrial melodism in the British pepper moth. So this approach, this framework from phenotypic differentiation to quantitative genetic approaches and genomic is what we have done in the Great Tit, and these are the results I'm going to present. But first, I want to finish this conclusion by telling you a little bit about a framework that is called the Pace of Life Syndrome framework. And mention that the work we've been doing was done in a context where we were particularly interested in studying a range of traits that might co-evolve in the urban environment. And within these traits, we are interested in personality and behavior traits. And in 2017, Sepp et al. Published a great review that provided some evidence that birds in the urban environment might be evolving toward slower paces of life. And this is based on a meta-analysis that they conducted on 15 avian species. And this meta-analysis showed in particular that urban birds lay smaller clutches and they have higher survival. And there are other traits as well, which are linked to this slower end of the pace of life continuum. So the birds have lower reproductive success and higher survival, but they also have weaker sexual selection and lower oxidative stress levels, for example. So we were quite keen to see where our great tits were standing in this framework. And the one thing we noticed is that in this review, Sepp et al. Did not take into account the work that had been done on behaviors. And there was actually, in the literature, there's actually quite a lot of evidence that urban birds are more aggressive and also that they show increased risk-taking behaviors. And these behaviors of higher boldness are usually associated with a faster pace of life. So we wanted to understand how this could fit in the pace of life framework. And I'll come back to this towards the very end of my talk. Okay, so I'm going to present now results from our Great Tit study. And the first thing I wanted to mention is that, as is the case for many empirical analysis based on long-term data sets, this is very much a collaborative project. And I want to especially thank Arnaud Grégoire and Marcel Lambrecht, who set up the nest boxes in 2010, and all the people that were involved in the field work. Most of the work that I will present today is based on stuff coming from Aude Kézer's PhD. Presently, she's in the second year of her PhD. And Charles Perrier, third year of PhD. And the work of the ERC-funded postdoc of Charles Perrier. We also have two external collaborators, Marta Sulkin in Warsaw and Juan Carlos Senar in Barcelona, who contributed to the axis on epigenomics that I will mention in the end. Okay, so the Great Tit project in Montpellier, as I said, the nest boxes were erected in the city in 2010, but we actually had a long-term study running since 1991 in the forest of La Rouvière, north of Montpellier here. And this picture here shows a snapshot of this forest, so a small part of the forest, where each dot is a nest box. And as you can see, it's a very natural oak forest with very little trails and roads. We have here 100 nest boxes for great tits and many more nest boxes for blue tits as well. In the city of Montpellier, we've also installed nest boxes in 8 different areas of the city. And there was a total of 250 nest boxes. Now, in the city, nest boxes tend to disappear, and so we have to set them up again. So this number tends to fluctuate over the years. And we're not winning the race, really. What was important for us is that we wanted to study great tits not only in urban parks, but also in very urbanized areas, because we had noticed that birds also breed in these very urbanized areas. So we really have a gradient of urbanization in the city and even within each site. So I mentioned we have 8 sites. This is a snapshot of one of our sites, and perhaps you don't see it clearly because it's too small, but you have an urban park here with different nest boxes, the small numbers, and there are also nest boxes in these streets with houses. So it means that even within a site, we had different types of urbanization. We measured this urbanization with what I will call the urbanization gradient. And this was measured at the level of each nest box. So around, within a 50-meter radius around each nest box, we measured vegetation cover, car traffic, pedestrian count, and the intensity of artificial light. And from this, we used a PCA, and it gives us an urban level for each nest box. Now, this graph here and the map here, they illustrate the average and variance of this urbanization gradient within each site and across the city. So as you can see, the forest here, Rouvière, is really very natural, but the more you go towards the south of the city, the more urbanized it is. And some of the sites like Gramont here, this GRA, they actually have quite a large variation in the urbanization around each nest box. So this is what we're going to exploit. So you might notice that there's actually only 5 sites here, not 8. This is because I'm presenting here, each dot is a blood sample that we have used for the genomic analysis I'm going to present, and this was based only on 5 sites. Okay, so a lot of us are ornithologists, even though I'm primarily an evolutionary biologist, but I don't measure only pedestrian and traffic disturbance. I also measure birds, of course. So additionally to the collection of the data that are characterizing the urban environment, we had quite a few measures on great tits. And these are really classical measures that you can find in any monitoring during the breeding season. So we capture birds when they breed, and we measure their morphology, especially their tarsus length, their body condition, and their wing and tail length relative to the body size. We also have measures on life history traits, such as the laying date, the clutch size, the number of hatchlings, and the number of fledglings. And then perhaps a little bit more original, we have behavior and physiological factors that we measure. So we measure handling aggression, which is a score given to each bird right after capture, depending on its reaction to the handler. We also have an exploration score, which is much trickier to measure. So for this exploration score, we use what is called an open field cage. The bird is placed in the cage here, and there's a camera at the other end that video records the behavior of the bird when it enters the novel environment that is the open field cage. And from the behavior of the bird, we measure an exploration score, which is based on the rapidity, the pace at which it explores this new environment, basically. And then we also have a measure of breath rate when we manipulate the bird, so under constraint. Okay, so we will start with investigating phenotypic variation between, so the divergence between forest and city birds, and also across the environmental gradient. And the summary I can give of these results is that pretty much any trait that we measured, all the traits that I presented, were significantly different between city and forest birds. And I'm presenting here means of just the dichotomy between forest and city birds. But if we when we look at gradient, we have also differences within the city, depending on the urbanization gradient. So birds in the city are smaller, they are lighter, and when we control for the overall body size, they have a smaller wing length and a smaller tail length. So this is for morphology. In terms of life history, birds in the city lay much earlier, actually 4 days earlier than birds in the forest. And they have a smaller clutch size, on average 2 eggs less than birds in the forest. They also have a smaller number of hatchlings and smaller number of fledglings. At the point when we published these results, this difference wasn't significant, but it now completely is. This shows that in fact 6 years of data wasn't enough to have a significant difference when we published. And in terms of physiology and behavior, birds in the city have a higher handling aggression score. Again, this is now significant with the latest data set. They are faster explorers when they are placed in the novel environment of the open field cage, and they have a higher breath rate under constraint. So I could mention, and I'm not going to show the result, that all these traits, for all these traits, we've also estimated repeatability, and for most of them, heritability. And all these traits are repeatable. And between our studies and other studies in other groups, we also know that all these traits are heritable. So the differences that we see between forest and city birds could well be a response to selection of these heritable traits, but it could also be due to plasticity. And this is one of the question that we want to answer. So is this difference a response to selection? And do we really have two ecotypes that have evolved over time? So the first step here are the selection analysis, and this is work that has been conducted by Odd Kezerg. So Odd has used the data that I have shown on all these traits and compare the phenotypic difference with the difference in fitness. And the first step was to use reproductive success as a fitness estimate. So what I'm going to do here before showing the selection gradients is each time I'm going to recall to you the main results in a simpler way. So birds are smaller in the city. Does it mean that selection is favoring smaller birds in the city? I'm going to walk you through this graph. This graph is showing linear selection gradients. When the linear selection gradient is positive, it means that reproductive success is higher for birds that have larger values for the traits. And when it's negative, it means that selection is favoring birds that have smaller values. When we are around 0 here, which is actually the case for most of these data points, it means that the selection acting on morphology is quite weak. You have in round the males, in triangles the females, and in purple the urban, and green the forest environments, the forest birds, sorry. So as I just mentioned now, overall what we find is that there is very weak selection on morphology, and the only two significant selection gradients that we found were showing selection favoring taller males in the forest and favoring lighter males in the forest. So we don't have any sign that selection is actually favoring smaller birds in the city, at least in terms of reproductive selection. In terms of life history, birds, as you may recall, lay earlier and smaller broods. So again, we wanted to check whether this was in line with the selection that we could measure, at least the selection acting now in the city and in the forest. And what we found is that if birds are laying earlier in the city, they are not selected to lay earlier in the city. They're actually selected to lay later, but this is not significant. They are selected to lay earlier in the forest. And if birds lay smaller broods in the city, they are in fact selected to lay larger broods. So birds that have larger broods have higher reproductive output. And then regarding behavioral traits and breath rate, this is a result that are still being worked by Odd Kezerg, and I'm not going to show you details, but what we find, and she has done here selection analysis with both reproduction and survival. And what she finds is that there is very little selection via reproduction again, but she also finds that all the behaviors that are displayed by urban birds, especially faster exploration, but also higher breath rate, they are all associated with lower survival in both habitats, in the forest and the city. So it doesn't seem that it's selected for in the city to have a faster exploration rate, or what I showed before. I probably shouldn't do that, but the fact that they have a faster exploration score, higher breath rate, and higher handling aggression is not aligned with selection favoring these traits. Okay, so what this means is that overall, the observed phenotypic divergence that I have illustrated before doesn't seem to be adaptive. It doesn't seem to be an adaptive response, at least not to the present reproductive selection, possibly not from what we can see in our latest results, to the present survival selection either. So it means that it is most likely a plastic maladaptive response, although we will still have to demonstrate this in the future. So the next step I had mentioned were experimental approaches. I'm not going to give any results here, although we have conducted experimental approaches to investigate the urban parameters that influence morphology and life history, but I don't have time to present this. I will say that next year, hopefully, we'll start a common garden experiment. So this is very exciting, but it's not stuff I can talk about today. And I will move on directly to this genomic approach where we have used rat sequencing, so what we have called reduction of the genome complexity, to explore how many genes and where are the genes that could be involved in urban adaptation. This is work that was conducted, as I said before, by Charmantier during his postdoc in my lab. So for this genomic and epigenomic axis of our work, I've highlighted here three main aims. The first aim we had was to test for neutral genomic divergence between urban and forest birds. The second aim was to test for genomic footprints of divergent selection between urban and forest population. And finally, we look at epigenomic basis of rapid adaptation, looking at differences in methylation. Okay, so the first axis is basically population genomics. And for this, we used 140 individuals that we blood sampled. So as I mentioned previously, this map here represents all the birds that we have sampled. And we sequenced them using rat sequencing. And what we obtained were 50,000 SNPs that were spread around across the whole genome. So that is after the filtering. Okay, the first graph I'm presenting here is the discriminant analysis of principal component, where each dot is an individual genome, and the closest to individual genomes are, the most similar they are. Most of you are probably very familiar with this. And what this DAPC shows is that if we represent the individuals by their sampling sites with different colors, we see that individuals are clustering within their sites, quite obviously. Now, this is in accordance with the FST. I don't think I'm going to show the FST table, but the FSTs that we found between the different sites of the city were all significant, but they were very small. And to highlight this, I've put here the strongest FST, the highest one, which is between the forest of La Rouvière and the most urbanized site of Masnouguier in the south of Montpellier. And this FST is of 1%. So for those of you that are familiar, this is quite weak a genetic differentiation. So we find weak but significant genetic differentiation across the city birds. And if we look at a redundancy analysis that investigates the simultaneous effect of latitude, longitude, and urbanization, what we find, and again, I'm sparing you the table, but we find that, of course, latitude and longitude matters a lot in explaining genomic differentiation, and you can see this in this DAPC graph here. But we also have an effect of urbanization at the site level. So I'm going to explain this here. When we feed in the RDA, the nest level urbanization, that is the level urbanization at 50 meters around each nest, this does not explain any genetic variance. But if we feed in the site level, so the average urbanization for all the nest box, say 1 kilometer around the focal individual, then it explains a significant proportion of the genomic variance. And this is 0.78% of the total variance. So I admit that it's quite small. But what is interesting is that it educates us on the scale at which urbanization impacts created population, because it means that what matters for the genomic trace of urbanization is not the urbanization 50 meters around the nest box, but rather more like 1 kilometer around the nest box. Another interesting result I want to present from this study is illustrated here by a map, and the same results are illustrated here in the graph. So on the top map here, you can see the urbanization gradient. Again, green is the least urbanized and red is the most urbanized. And you can see that the more you go towards urbanized, highly urbanized area, the smaller the heterozygosity levels are, because you go from blue, high heterozygosity, to red, which is low heterozygosity. It's exactly the same thing for those of you that are more keen on graphs that you can see on this graph here. And the fact that individual heterozygosity are lower in more urbanized sites means that population sizes are probably smaller, or populations are less connected in more urbanized sites. Okay, so the second aim we had in this genomic approach was to test for genomic footprints of divergent selection. And I need to go back a step here, because I have mentioned that we believe, after the study we have done on selection, that the phenotypic divergence that we see is probably due to plasticity, but it doesn't mean that these traits or other traits have evolved and adapted in the urban environment. And this is what we're investigating here. So here is the Manhattan plot, which I will call a Montpellier plot, because it's showing Z-scores for the strength of the association between each SNP variation across the genome and variation in urbanization at the site level. Results are actually very similar if we use urbanization at the nest box level. So the reason I'm saying it's not really a Manhattan plot, it's because there are no large Z-score peaks. What we do find are several SNPs with relatively high scores of associated that are highlighted here in red. And a top subset of 97 of these SNPs associated with urbanization explain 81% of the urbanization score. So overall, this means that there is no huge, strong SNP associated with urbanization, but there are numerous SNPs that have small effects across the whole genome. This is indicative of polygenic adaptation, which we could have expected. And it's actually similar to what has been found for traits such as phenology or bill length in the gray tits and in other birds as well. So selection induced by urbanization is most probably a complex process, and it involves very different traits. And we think, of course, that many of these traits are probably quantitative. So this means that polygenic adaptation is something that we could have expected. Now, I'm not going to show details on the gene ontology, but what we have as result, and this is in the paper of Schall, published in 2018, is that the gene ontology shows enrichment of neuronal function. We think this is something that is very exciting because it might be particularly linked to the personality variants that we see, or other behavioral traits that differ between urban and forest birds quite classically. Okay, so the last set of results regards epigenomics. So in the literature, there is growing evidence suggesting that epigenetic mechanisms such as the DNA methylation that I illustrate here may be involved in rapid adaptation to new environments. And in particular, air pollution has been demonstrated to cause DNA methylation change, most notably in blood samples in humans, but also in non-human animals. So what we did here is we targeted a study on methylation using samples from 3 different cities. And so this is the collaboration we have with Marta Solkin in Warsaw and Juan Carlos Senar in Barcelona. And we added samples from Montpellier. So we had 20 blood samples per site, 10 urban, per location, 10 urban and 10 forests. But in fact, because in Montpellier we have a diversity of landscapes, we took 20 birds from the urban Montpellier area. This is why we have 70 and not 60 individuals overall. And with these 70 individual blood samples, we conducted RAD sequencing to look at the whole genome differentiation, pretty much like we did in the study that I presented earlier in Montpellier. And we studied methylation using reduced representation bisulfite sequencing, or RBS. So we had 80,000 SNPs and many, many sites for the RRBS analysis. And I'm going to show these 2 steps separately. So I will first show the RAD sequencing results and then the RRBS results. So with the RAD sequencing, we obtained FSTs between the different city, urban, and forest sites. So here in diagonal and in color, you have the FST for Barcelona urban versus forest, Montpellier urban versus forest, and Warsaw urban versus forest. And as you can see, a lot of these FSTs are actually significant. I think all the ones I'm presenting here are significant, but they are quite small, similarly to what we showed previously. the highest FST we found was between birds from the Barcelona forest population and birds from the Warsaw forest population, and this FST is of 2.5. This is to give you an idea of the level for people that are familiar with these FST measures. So overall, this means that there is genetic structure across these populations, but it's quite weak, in contrast with previous results that had been found on using microsatellites, which showed quite larger FSTs. Now, since we know, we actually think that this context is quite ideal to explore epigenomic footprints, because our FST results show that the structuration across the whole genome is quite weak. So if we were to find strong epigenomic footprints of urbanization, it could be attributed to, it could not be attributed to strong background neutral genetic divergence. So we have low genetic divergence in the background, and that means that we might see epigenomic footprints more easily. So staying with the RATSEC, I'm talking about epigenomics, but we're not there yet. Staying with the RATSEC results, I wanted to present the RDA results here. This is the illustration. What we found was that what explained genomic variance across for all the birds in the 3 different cities is first-hand latitude and longitude, so geographical distance. And you can see here the RDA axis 1 is here, and you can see that it differentiates individuals sampled in Barcelona in orange, with individuals sampled in Warsaw in yellow, and individuals sampled in Montpellier in purple. But the third axis of variation is this urban-rural contrast. And you can see this as well quite clearly here, because on the bottom size of this RDA3, you have all the urban individuals, and on the top, you have all the rural individuals. So you have a clear contrast, for example, here in Barcelona, between forest and city in full dots, and empty dots are the forest birds. And then I'm mentioning here, because it's important for the comparison with the epigenomic results, the fourth axis of variation was sex. So we know, of course, that across the whole genome, there are very often differences between males and females, but this was much weaker than the differences that we could see across the geography or between urban and rural contexts. I actually wrote rural here, but I really want to mean forest. I should delete all these rural mentions. Okay, so now I'm moving on to the epigenomic variation and the similar RDA analysis on the epigenome. Unfortunately, these are results that, well, not unfortunately, but these are results that are very, very new. Aude Kayser, my PhD student, is still working on them with Charles and other collaborators. And so I don't have this similar graph for the RDA, and I don't want to show you the table that is much too complex. But what I will say is that the first axis of the first factor that explains epigenomic variance is sex. And there's a very strong contrast between epigenomic marks between females and males. The second axis is geography, it's latitude and longitude. And this third axis is the urban-rural contrast. And this, the urban origin of birds, or the forest origin of birds, explain 1.6% of the variance, of the epigenomic variance. So I think I forgot to mention here somewhere, that for the rat sequencing, so the whole the approach across the whole genome, we had 2% of the variance explained by this urban-rural contrast. And here for the epigenomic level, we have 1.6% of the variance, and it's not quite significant. So we are running analysis right now as I'm talking. 2 offices behind me, there's a computer working on this, but the first results that I'm showing here in a qualitative way seem to show that there is no strong evidence for epigenomic marks of urban adaptation. I do want to show you this really cool circus plot, even though it's very preliminary. So Aude told me it's probably going to change, perhaps drastically, but the first plot that we got is very interesting because it illustrates what I just said. So this circus plot represents the graded genome. You have here, of course, you probably can't see, well, I can't see, but chromosome 1, 1A, 2, 3, etc. And you have the red bars. I hope that you can see them. They represent differentially methylated regions, so DMRs, where there is hypermetylation in cities. And the blue bars represent areas, DMRs with hypomethylation. So the first thing we can notice is that there doesn't seem to be more hyper than hypomethylation in city. We could have expected that pollution and all the other characteristics of the urban environments will induce hypermethylation, but it doesn't seem to be the case. At least there are also a lot of hypomethylations. And then I wanted to illustrate this contrast between males and females. Here you have the surplus plus for females in pink and males in blue. We're not being very original. And you might be able to notice that many of the areas where you have, for example, hypomethylation in female, you don't have the same in males. So there's very strong differences between males and females in their methylation contrast between forest and city. So what we're doing now is we are finalizing these analyses. We especially want to investigate parallelism across the 3 cities, Montpellier, Barcelona, and Warsaw. And at first hand, it seems that methylated regions are actually not the same in different cities. So you would find circle plots to be very different across these 3 cities. And also we want to explore whether where the DMRs are located in the genomes, whether it's in exons, introns, or in non-coding regions. And on this, I don't have any answer yet, but hopefully in a short while time. So I will conclude by saying that overall, we have seen strong phenotypic differentiation that does not seem to be explained by selection acting presently. We have shown that there are small effects of urbanization on the genetic structure, and there is an effect of urbanization on the genetic and epigenetic diversity, with mostly a lot of small effects, so a polygenic adaptation to urbanization probably. And I wanted to finish with this idea of pace of life. So I've pasted here the framework figure from the seminal paper of Denis Real et al. In 2010 in Filtrans, where they postulate that populations that experience different ecological conditions should differ in a correlated suite of traits. And that means that individuals would range, or populations, from the fast end to the slow end of the pace of life continuum. And what we wanted to know is where our great tits were standing here. What we have found, we haven't found, as Sepp et al. Have found in their analysis, we haven't found a slower pace of life in the city. We had found, on the contrary, that birds seem to display traits that are associated with a faster pace of life. Especially, they have higher aggressiveness, they are bolder, they explore their environment more quickly, and they have higher breath rate, which is associated with a high sympathetic system reactivity. We are presently also running survival analysis, and the results that I don't have time to detail don't seem to point towards slower, longer survival, longer lifespan in the city. If anything, they have smaller survival, but this is really not significant. So we don't see any difference in survival between forest and city birds. So what we want to do now is we want to complete this picture, and we want to study fully survival, but also dispersal, cognition abilities, oxidative stress, immune response. This is our future, and it will be done across France, across 5 cities in France, and this thanks to funding that we just acquired. So this might be the next story that I will tell you, and I am gonna end here and answer some questions. Thank you very much for listening.
Thank you very much, Anne, for a fascinating talk, a huge amount of work, and yeah, really nice presentation. Thank you. We do have questions coming in, and there's more coming in as we speak. So the first question is, the phenotype you describe are very similar to what one sees when birds colonize islands. What would you hypothesize regarding phenotype and urban environments on islands? Would the difference be even more than what you see in your studies, shorter tails and wings, and even more bold?
So that's actually an interesting question, because before studying great tits, I studied blue tits, and especially blue tits in Corsica, which is an island. And we've studied, all these traits I'm showing here, we've studied them in blue tits, and they do in fact have similar types of phenotypic divergence between the island of Corsica and the mainland. And Corsica is quite a large island, but we still find this insular syndrome in morphology, but also in personality traits, which is interesting. I don't know of any, there are probably, but I don't know of any study that have looked in details at cities in islands. Well, there are studies in the Galapagos where they have seen, similar differences in cognition and behaviors between birds that are in cities and outside cities within the island, but I don't think they compared birds between the island and the mainland. It's an interesting question, but yeah, I don't have more than that to say. We can compare, in many ways, we can compare cities to islands probably. So it's, yeah, thank you for the suggestion that I should link the two fields.
I guess, yet more work than to look at cities and urban environments on islands versus mainlands. So I think that's really cool though. It's starting to come together, and as you said, like there's so many different sites where one can collaboratively, I guess, look at these questions. So the next question from audience is, do you see sex differences in phenotypic response to urban environments?
So I think the graphs I've shown you here don't show sex-specific differences. Let me just check, because we have them. No. No, we don't see very strong. Overall, both males and females are smaller and have shorter wings and tails, etc. But we are hoping that we could look at dimorphism. We had a student that looked at the sexual dimorphism, and presently we don't have any strong results. It doesn't seem that dimorphism is changing. But one thing that I haven't shown here that might give a different answer is that we're also studying the size of the breast patch in great tits, the black, which is a sexual ornament. And here we might have different results between males and females, but it's an ongoing project.
Cool.
So can I just add, if the person asking the question knows of any theoretical work that would suggest that the response of females and males should be different, I would be very interested to have this knowledge.
Mm-hmm. I guess maybe it ties in with the fact that there seemed to be at least some indication that the selection coefficient was kind of opposing for males at least. So if there was the selective response, I guess maybe you would.
Yeah, I understand where you're coming from. Yeah, that's right. That the selection pressures aren't the same for males and females, certainly. So you could expect if some of these traits have a genetic determinism that is sex-specific, it might evolve differently in males and females. But I'd say for most of these traits, there's a very strong cross-sex genetic correlation that would constrain at least rapid evolution in different directions.
Sure. Right. So next question is, do you have a rough estimate on how many generations the urban birds have been urban birds, and how much urban birds become rural and vice versa? As in, is there migration?
Yeah, so the question about how old our city is very important, I think. When we are thinking about comparing across city, one of the interesting things would be to look at cities or areas that are less ancient. Montpellier is a very old city. It's one of the first universities, especially the medical university is 1,000 years old. So this gives an idea of how old the city is. And a great tit generation is around 2 years. So that would mean we have at least 500 generations of great tits in the city, probably because we know that they've been around probably forever. Now, the second part of the question was, can you remind me?
How much gene flow?
The gene flow. So from the FSTs that we've estimated, we think there is gene flow. But one of the areas that I would like to explore more in the future are suburban areas. I hope that's how you say it. So these areas that are in between the city and the forest, because this is probably the area where there's the highest variation in genetic backgrounds and where the gene flow is happening. So yeah, the FSTs we have seem to suggest that there is gene flow. What we haven't been able to get out yet is whether this gene flow is unidirectional, because there are, hypotheses in the literature about the source sink dynamic that might happen in the city, and we don't have an answer to that yet.
So I'll just go to the next question of the audience. I was wondering if there are genes associated with song adaptation to urban environments. Is there a behavioral change in that that you know about?
Yeah, so we haven't looked at it, but there's quite a few studies that have shown, I don't have the English names in mind, but in the blackbird and other species, that there is song adaptation. Is there one in great tits? I'm not sure now, but yeah, there are quite a few people that are looking at song. We don't have any data in our own study about, so the results are usually that they have a higher pitch tone and that they sing more loudly in the city, but we don't have any data on this. There are people in Paris, I know, that are working on this and how this affects communication between parents as well for raising their chicks, but I don't have any data in my own study.
Wonder if the same happens in humans.
We talk louder when we're in a crowd, definitely.
or faster, anyways. I think from Switzerland, there's that stereotype that I know.
I don't know if it has reached genetic adaptation, though.
Yeah, no, probably not. It's probably plastic, like you say. Right. Okay. I'm just going through the question. We probably won't have time to cover them all, but Anna's kindly offered to go through questions later and see if she can answer some more. Have you looked at resource availability and how that affects the abundance across the urbanization gradient?
Yes, thank you. Or should I say, you shouldn't have asked this question. This is the trickiest question. So the experiments that I didn't mention, I said, or that I didn't explain, I said that we have conducted experiments. And these experiments were mainly to test the idea that the fact that these birds are smaller and lighter is due to the fact that they don't have enough resources. It is very tricky to demonstrate. It's very tricky to have data. I said before that it's actually very easy in the city to have a whole lot of environmental features, but the abundance of insects and caterpillars in particular, it's not very easy to access. So we performed experiments where we did two kinds of experiments working on the size of the broods and also a supplemented feeding experiment. It didn't work out the way that we expected. But there's a paper that just came out from another group where they do show that when you supplement birds with caterpillars outside their nests, it increases the number of offspring raised and the weight of the offspring. So yeah, I'm quite convinced, at least intuitively, that low food resources is one of the problems leading to this smaller size in the city.
So I mean, the fact that you could run a very neat PCA on these different characters, I guess, suggests that they're always quite strongly correlated, or is there enough variation maybe between cities that, I know, in the longer run, one might get an idea of which of the urbanization factors are more causal?
Well, this is the reason why we decided that we were setting nest boxes in areas that are parks, but also in very urbanized area within the same site. And we think that, we have a pretty good data set to decipher between vegetation cover, which is our proxy of food abundance, basically, and things like a pedestrian or traffic. We have areas, for example, in the university, there's a lot of pedestrian disturbance, but there's no traffic. And there's not a lot of vegetation. In other areas like the zoo, there's a lot of vegetation, a lot of people as well, but no traffic at all. And then in other areas, you have the whole set of disturbance. So unfortunately, for the time being, this is quite an early project. It's quite recent. It's not very old. So we have mainly used this PCA and the urban gradient. We haven't looked precisely into the different parameters, but I think we're reaching the 10th year of the project. So if anyone wants to work on this, I'm quite happy to start now, looking more deeply into each parameter and whether we can decipher the different effects.
Very cool. Right, I think in the sake of time, we'll leave it for now. There are more questions, so when you do get the time, please do go and check them out if you can. Thanks
I
again.
will do. Just don't expect me
No, sure.
this
There's
evening. I'm just telling people, do come back on Monday because I will take the time over the weekend. But yeah, I will do, and I'm very happy for this opportunity to prolong the discussion with you guys. Thank you.
Thank you very much. Thanks again for a great talk. And
thank you for inviting me. I'm very flattered. Thank you.
All right. So just a short announcement before we go. We have 2 more weeks where we have 3 talks on Monday, Wednesday, and Friday. Before then, in July and August, we'll reduce a bit. We'll have 1 talk per week on Wednesdays. But yeah, stay tuned. Check out our, follow us on Twitter, Evo Eco Seminar, sorry, and join our Slack channel to keep up to date with what's going on. Our next talk will be this coming Monday. Dr. Lucy Aplin will talk about animal culture in a changing world, from diffusion of innovation to cultural evolution in birds. So not that dissimilar from our talk today.
You're going to hear about great tits again.
I don't know what species, to be honest. Right, so with that, we're at the end of the talk. Thanks all for listening, and see you again on Monday. Bye.
Thank you, bye-bye.